In this video tutorial, I am going to discuss about regulation of glycolysis. In one of my previous video of this playlist, I had already discussed about all the reactions of glycolysis. So these are all the reactions of the glycolysis, and we know that all the reactions of glycolysis, they are reversible except for the three reactions which are irreversible. And in this diagram, all three irreversible reactions are shown by red arrow. The first irreversible reaction is this reaction, which is catalyzed by the hexokinase or glucokinase. The second irreversible reaction is this reaction, which is catalyzed by the phosphofructokinase 1 enzyme. And the third and final irreversible reaction is this reaction, which is catalyzed by the pyruvate kinase. Regulation of glycolysis occurs at all these three irreversible reactions, but the most effective and most prominent regulation occurs at the second irreversible step. Now, why this second irreversible step is most important regulatory point for the glycolysis? The reason is that this is the slowest step of the glycolysis. So this step determines the rate of entire glycolytic pathway, right? So because of it is slowest, it becomes a rate limiting step. So if you manipulate the speed of this step, then the speed of entire glycolytic pathway will be effectively manipulated. And the second reason is that this is a committed step. This is the committed step. That means, see, once fructose 1,6-bisphosphate is formed in this second irreversible step, there is no any other fate for the fructose 1,6-bisphosphate other than the glycolysis. It has to undergo glycolysis. So there is a commitment towards this glycolytic step. See, here glucose 6-phosphate is formed. This glucose 6-phosphate is not committed to glycolysis. Why? Because of course it can undergo glycolysis, but it can undergo some alternative pathway. For example, glycogen synthesis. It may undergo HMP pathway, that is hexose monophosphate pathway. So there is no commitment for the glycolysis over here, whereas here there is a commitment towards the glycolysis. Right. So because of these two reasons, this second irreversible step is going to be the most effective regulatory point in case of glycolysis. So let's look at the regulation of glycolysis at all these three irreversible steps. We will start with the first irreversible reaction. So the first regulatory point for the glycolysis lies in the glucokinase or hexokinase. What this glucokinase or hexokinase does is that first glucose is converted to glucose 6-phosphate, right? And this reaction is catalyzed by the hexokinase or glucokinase enzyme. Now remember, hexokinase and glucokinase both are isoenzyme. That means they catalyze the same reaction, but their kinetic properties are different. And because of the difference in the kinetic properties, their entire role in the glucose metabolism is different. I had already discussed this different role of hexokinase and glucokinase based on their differences in the kinetic property. I had already discussed, so I am not repeating that. Rather, here we will focus just on the regulatory aspect of these two enzymes. Now, hexokinase and glucokinase both are differently regulated. Okay, let's first discuss about hexokinase. This hexokinase is allosterically inhibited by this glucose 6-phosphate. So glucose 6-phosphate, it allosterically, so allosteric inhibition is there for the hexokinase. So by this inhibition, it is our cell is making sure that glucose 6-phosphate is not generated more than the cell can utilize, right? See, if glucose 6-phosphate is generated more, then what will happen? It will immediately inhibit this hexokinase. So glucose will stop producing glucose 6-phosphate, and so glucose 6-phosphate concentration inside the cell will always under a check. How glucokinase is getting regulated? One important aspect is that glucokinase is not inhibited by its product glucose 6-phosphate. Rather, glucose 6-phosphate is reversibly converted into the fructose 6-phosphate. This is the second step of glycolysis, right? So this fructose 6-phosphate, it indirectly inhibit this glucokinase. So this inhibition is indirect. It indirectly inhibits glucokinase. Whereas this glucokinase, which is the substrate of glucokinase, it indirectly stimulates this glucokinase. So this stimulation is also indirect. Now why I had written indirect over here? How actually this fructose 6-phosphate inhibits glucokinase and glucose stimulates glucokinase? Let's see. Suppose this is the glucokinase, right? Now let's say this is the active site of glucokinase. Now whenever glucokinase is free like this, it is in the active form. That means it can convert glucose to glucose 6-phosphate. But in our cell there is one more protein which is known as Glucokinase regulatory protein, that is GKRP. Now GKRP has a binding site over here which can bind with the active site of glucokinase. So both can undergo reversible binding. So this is the reversible binding with glucokinase and GKRP. So this is the associated product, that is glucokinase, its active site is over here. This is the binding site of GKRP, so binding site, and this GKRP is bound over here. So as this glucokinase regulatory protein is binding with the glucokinase at the active site, this glucokinase is inactive. And the second aspect is that this GKRP, when it binds with the glucokinase, it translocates to the nucleus. So translocated to nucleus. And remember, glycolysis is the cytoplasmic process. So once this GKRP anchors glucokinase inside the nucleus, what will happen? Glucokinase is cleared from the cytoplasm, and so glucokinase can no more active. So here two-way effect is that. First, this GKRP makes glucokinase inactive. Second, it is translocated to the nucleus, right? So this way glucokinase becomes inactive. So this association, or you can say inactivation of glucokinase, inactivation of the glucokinase is carried out by the fructose 6-phosphate. So what fructose 6-phosphate does? It makes glucokinase and GKRP binding. So it stimulates this association, and by this it can inhibit glucokinase. That's why I had written over indirect inhibition of the glucokinase by the fructose 6-phosphate. What glucose does? So glucose, what it does is it competes with this GKRP for the binding with the glucokinase. So if you provide more glucose, then it stimulates dissociation of this complex and make glucokinase active, and that's why I had written over here that glucose indirectly stimulates glucokinase, that you can see from this diagram. Okay, so this is the regulation at the glucokinase hexokinase level. Now next see about the regulation of glycolysis at this second irreversible step, which is catalyzed by the phosphofructokinase. So second is phosphofructokinase 1. Now this regulation is very important. In this step, what is happening? The fructose 6-phosphate is converted to fructose 1,6-bisphosphate. So fructose 6-phosphate is converted to fructose 1,6-bisphosphate, and then it undergo glycolysis. It follows the steps of glycolysis. This conversion of fructose 6-phosphate to fructose 1,6-bisphosphate, it is catalyzed by the phosphofructokinase 1 enzyme, and here ATP is converted to the ADP, right? Now this phosphofructokinase 1 has a several allosteric sites, and by that allosteric site it is allosterically either stimulated or inhibited. So the first important allosteric site is for the ATP. ATP inhibits phosphofructokinase 1, and another molecule, that is citrate, it also inhibits phosphofructokinase 1. Remember, these both inhibition are the allosteric inhibition. Okay, two more molecules are there, ADP and AMP. This both molecule allosterically stimulates phosphofructokinase 1. Now what is the logic behind all these four molecules? Some of them are stimulating, some of them are inhibiting this phosphofructokinase 1. Let's try to understand. See, whenever ATP is there, ATP in the enough concentration, it suggests that cell has enough amount of energy. It is suggesting of energy energy rich state of the cell. So when energy is in enough amount, glycolysis should not occur. Why? Because glycolysis, its main purpose is to provide energy, and it will be useless to get more energy when you have enough energy. So ATP, high concentration of ATP, it inhibits phosphofructokinase 1. It makes sure that when enough amount of energy or cell is in the energy rich state, glycolysis should not occur, and that's why phosphofructokinase 1 is inhibited. Similarly, citrate, it is also inhibiting phosphofructokinase 1. But why citrate is inhibiting phosphofructokinase 1? What is the purpose? See, citrate, it is the key intermediate for the energy metabolism of the pyruvate, fatty acid, and some of the amino acid. All these molecules energy metabolism, citrate acts as an intermediate. So when we have enough amount of citrate, it is indirect indication that enough amount of this foodstuff of this molecule are undergoing energy metabolism. So citrate is also suggesting an energy rich state. Okay, and that's why it is inhibiting phosphofructokinase 1 and making sure that no glycolysis occur in energy rich state. Now what about ADP and AMP? See, ADP and AMP both are a broken down product of the ATP. So when ATP loses its energy, ADP and AMP are generated. So both of these molecules, they are suggesting of energy poor state of the cell, and that's why when energy is not there, our body needs glycolysis for the energy purpose, and that's why by stimulating phosphofructokinase 1, it makes sure that glycolysis occurs. Now at this point, I would like to point out one more thing, that conversion of fructose 6-phosphate to fructose 1,6-phosphate, it leads to glycolysis, but the reverse reaction is also possible by one more different enzyme. This fructose 6-phosphate, it can be converted to fructose 6-phosphate by one different enzyme, that is fructose 1,6-bisphosphatase. Okay, what this enzyme does, this enzyme adds water molecule and remove phosphate. Now what is the role of fructose 1,6-bisphosphate? Fructose 1,6-bisphosphate is causing a reversal of glycolysis, and reversal of glycolysis is the gluconeogenesis. So when fructose 1,6-bisphosphate is working, what will happen? Fructose 1,6-bisphosphate will be converted to fructose 6-phosphate, and then by the several reversible reaction, it ultimately generate glucose. And this step, this side of reaction, we call it as a gluconeogenesis. Gluconeogenesis. Neo means new and gluco means glucose. So new glucose will be generated. See, this is the opposite pathway of the glycolysis. Why? In the glycolysis, there is a breaking down of the glucose. In the gluconeogenesis, it is a new generation of glucose, right? So in our cell, whenever glycolysis is occurring, gluconeogenesis should be stopped, and whenever gluconeogenesis is occurring, glycolysis should be stopped. There is no way these two pathways can be active simultaneously. Why? Because then it will become a futile cycle. That means our body is continuously synthesizing glucose and continuously breaking down of the glucose, right? So if both pathways occur simultaneously, it is simply a wastage of our cell's energy. That's why it is called a futile cycle. So in the nature, it is logical that whenever glycolysis is occurring, gluconeogenesis should not occur, and whenever gluconeogenesis is occurring, glycolysis should not occur, right? So that means both pathway should be reciprocally active, right? So how it is reciprocally regulated? So here this AMP molecule, it is very potent inhibitor of this fructose 1,6-bisphosphatase. This AMP molecule will make sure that when glycolysis is occurring, gluconeogenesis should be stopped. Now, apart from this four molecule, there is a fifth molecule which is very important regulator of both this phosphofructokinase 1 and fructose 1,6-bisphosphatase. Actually, I had kept the best molecule for the last, and that is fructose 2,6-bisphosphate. This fructose 2,6-bisphosphate, it is the most potent activator of this phosphofructokinase 1, and it is the most potent inhibitor of fructose 1,6-bisphosphate. It is said that in the absence of fructose 2,6-bisphosphate, phosphofructokinase activity is near to the zero. It is near to the inactive, right? So let's discuss about this fructose 2,6 bisphosphate. Now from where does this fructose 2,6 bisphosphate is coming? Surprisingly, it is synthesized from this fructose 6 phosphate. This fructose 6 phosphate gives rise to this fructose 2,6 bisphosphate, and here the enzyme is phosphofructokinase 2. So we should note that this phosphofructokinase 1 and this phosphofructokinase 2 both are similarly looking but totally different enzyme, right? And here meanwhile it utilizes ATP. ATP is converted to ADP because one phosphate is more than the fructose 6 phosphate, right? Then how fructose 2,6 bisphosphate is removed? Fructose 2,6 bisphosphate is broken down by removal of phosphate to the fructose 6 phosphate. So here water molecule is added and one inorganic phosphate is removed, and the enzyme which is working over here is fructose 2,6 bisphosphatase. So here also we should remember that this fructose 1,6 bisphosphatase, it is different from this fructose 2,6 bisphosphatase. Both are totally different enzyme. Now there is one very important aspect about this phosphofructokinase 2 and fructose 2,6 bisphosphatase. See, phosphofructokinase 2, this phosphofructokinase 2 and fructose 2,6 bisphosphatase, both are of course a different activity, but both are part of one single protein. So this both activity lies on one single protein. So this is a single protein having a two functions, right? Having a two function. One function is phosphofructokinase 2 activity and another function is fructose 2,6 bisphosphatase activity. So we can call this as a bifunctional enzyme. Bifunctional enzyme. Now luckily what happens is that whenever phosphofructokinase 2 is active, fructose 2,6 bisphosphatase is inactive, and whenever fructose 2,6 bisphosphatase is active, phosphofructokinase 2 is inactive. Now what determines that which function will be carried out, right? So but before that let's write this complete structure at both this place. So here phosphofructokinase 2 is active and fructose 2,6 bisphosphatase is inactive, and this is the single protein molecule. Similarly, here fructose 2,6 bisphosphatase is active, but phosphofructokinase 2 is inactive, and this is the single molecule of protein, right? So as we can see here, phosphofructokinase 2 is active, but fructose 2,6 bisphosphatase is inactive, and here phosphofructokinase 2 is inactive and fructose 2,6 bisphosphatase is active. Now what will determine that which action or which function will be active? So that is determined by the phosphate group. If phosphate group is present, if this protein is phosphorylated, then it says that fructose 2,6 bisphosphatase is active and phosphofructokinase 2 is inactive. Whenever phosphate group is removed, that means dephosphorylated state, it suggests that phosphofructokinase 2 activity is there and fructose 2,6 bisphosphatase is inactive. Now how this phosphorylation and dephosphorylation goes on? So this gets phosphorylated by one enzyme. Its name is cyclic AMP dependent protein kinase. This cyclic AMP dependent protein kinase, it phosphorylates this bifunctional enzyme. So fructose 2,6 bisphosphatase will be active, right? Now this cyclic AMP dependent protein kinase, it activated in the presence of cyclic AMP, and this cyclic AMP is stimulated, its production is stimulated by the hormone glucagon. Okay, I should use red color. So glucagon, it stimulates this cyclic AMP, and this cyclic AMP in turn stimulates cyclic AMP dependent protein kinase. How it is getting dephosphorylated? So dephosphorylation is carried out by one enzyme, that is protein kinase. Protein kinase. Now this protein kinase is under the influence of insulin. Presence of insulin stimulates protein kinase enzyme, and so it gets dephosphorylated, right? So see what happens whenever you are in the fasting. So what will happen? And yes, I forgot to mention one thing, that this regulation by the fructose 2,6 bisphosphate, it occurs in liver only. Okay, in liver only. So whenever you are fasting, what happens? There is a low glucose concentration in the blood. So low glucose concentration, it stimulates glucagon. So now glucagon, which appears during the low glucose concentration, it causes the phosphorylation of this protein, and so fructose 2,6 bisphosphatase activity is there. And what this fructose 2,6 bisphosphatase will do? It will decrease the concentration of fructose 2,6 bisphosphate. So what will happen? As fructose 2,6 bisphosphate is stimulating the glycolysis, less fructose 2,6 bisphosphate will inhibit. So there will be a less glycolysis. Whereas this fructose 2,6 bisphosphate, as it is inhibiting fructose 1,6 bisphosphatase, so in this case what will happen? It will stimulate gluconeogenesis. And remember, this both action is occurring only in the liver. So during the fasting state, liver stop utilizing glucose, rather liver starts generating glucose. What will be the purpose? This generated glucose will diffuse into the blood and it will reach to our essential organ like a brain and other tissue. So it spares glucose for the utilization by other tissue. So this is the impact of this regulation, right? Now there is one very important molecule which can influence this protein kinase. It is the xylulose 5-phosphate. This xylulose 5-phosphate, it stimulates protein kinase. Now why this is very important molecule? Because this xylulose 5-phosphate, it links carbohydrate metabolism with the lipid metabolism. How? See, what happens? Whenever there is high glucose concentration, it leads to stimulation of all the different glucose utilizing pathway, and one of the glucose utilizing pathway is the hexose monophosphate pathway. The hexose monophosphate pathway, xylulose 5-phosphate is one of the important intermediate of this hexose monophosphate, and apart from that, NADPH is one of the important product of this HMP pathway. This increased glucose, of course, it can undergo glycolysis also, and by the glycolysis, it can generate pyruvate. And this pyruvate, it can generate more and more acetyl coenzyme A. And see, this xylulose 5-phosphate, it is also stimulating this glycolysis, as we had just seen, right? If xylulose 5-phosphate, more of this fructose phosphofructokinase 2 activity, more fructose 2,6 bisphosphate, more action of phosphofructokinase 1, and so more glycolysis. So it produces more and more acetyl CoA. So this NADPH and this acetyl CoA both are the starting material for the fatty acid synthesis. Right? So, and one more thing, that xylulose 5-phosphate is known to stimulate all enzymes of this fatty acid synthesis. So what happens whenever there is high glucose concentration, it leads to high fatty acid synthesis. So it is generally said that high carbohydrate diet can lead to high fat content in our system. Okay, so that linking point is created by xylulose 5-phosphate. So that's all discussion about this second irreversible step, that is phosphofructokinase 1. Let's discuss about this third and final irreversible step, that is pyruvate kinase. How pyruvate kinase is regulated. So third irreversible step, which is catalyzed by the pyruvate kinase, it converts phosphoenol pyruvate to the pyruvate, and this reaction is catalyzed by the pyruvate kinase. In our body, there are two different types of pyruvate kinase. One is pyruvate kinase L and second one is pyruvate kinase M. This pyruvate kinase L is found only in the liver, whereas pyruvate kinase M, it is found elsewhere and especially in the skeletal muscle. Okay. So, both of these isoenzymes, they carry out the same reaction, conversion of phosphoenol pyruvate to the pyruvate. And we know that phosphoenol pyruvate, by the multiple reversible step, it is generated from the fructose 1,6 bisphosphate, which is the product of our second irreversible step, right, from the fructose 6 phosphate. And this enzyme is phosphofructokinase 1. Now, what happens? This pyruvate kinase L and M, they are allosterically stimulated by this fructose 1,6 bisphosphate. Now, what is the importance of this stimulation? See, this is fructose 1,6 bisphosphate, it is the product of phosphofructokinase 1, and its immediate product is stimulating the FAR kinase reaction, right? So, it is essentially linking this kinase with this kinase. Okay. Second is that this pyruvate has a multiple fate. Pyruvate can get converted to acetyl-CoA, right? And this acetyl-CoA undergo TCA cycle to produce ATP, or it can be used as a starting material for the fatty acid synthesis. So, fatty acid may be synthesized from this acetyl-CoA, or this pyruvate can undergo transamination reaction to give rise alanine. Okay. So, all these things, extra ATP, fatty acid, and especially long chain fatty acid, which suggest more and more acetyl-CoA is present, more and more alanine, all these things, they inhibit this pyruvate kinase. So, pyruvate kinase is allosterically inhibited by the acetyl-CoA, this acetyl-CoA, it can be inhibited by the ATP, it can be inhibited by this long chain fatty acid, or it can be inhibited by the alanine. So, inhibition by all these things, it will make sure that pyruvate is not generated more than the cell can utilize. Okay. So, always pyruvate concentration will be always under a check. So, by this way, both of this type, L type and M type of pyruvate kinase are getting regulated. But there is something more to the story for the especially pyruvate kinase of L type. Remember, L type is present only in the liver. What happens is that this pyruvate kinase L type, it is regulated by the covalent modification of the protein by the phosphorylation or dephosphorylation. Whenever phosphate is not there in this pyruvate kinase structure, it is active. Whereas, whenever the pyruvate kinase L type has a phosphate, or you can say it is phosphorylated, it is inactive. So, what causes the dephosphorylation and phosphorylation of pyruvate kinase type L? Remember, this is true only for the liver type. M type is not regulated by the phosphorylation and dephosphorylation. So, pyruvate kinase L, it gets phosphorylated by one enzyme, that is cyclic AMP dependent protein kinase. Right here, ATP is utilized for the donation of phosphate to this protein. So, ADP is generated. Right. Now, this is getting stimulated by the cyclic AMP, and this cyclic AMP, as we had discussed earlier, it is synthesized, its synthesis increase in the presence of glucagon. So, whenever glucagon is more and more this pyruvate kinase undergo phosphorylation, and so this glycolysis is inhibited. Okay. But where is it inhibited? It is inhibited only in the liver, remember, not in the muscle. Okay. Now, how this pyruvate kinase L gets dephosphorylated? So, again the enzyme is protein phosphatase. Protein phosphatase, this enzyme causes the removal of phosphate. Okay. Here water molecule is added and inorganic phosphate is removed. Now, see what will happen, what will be the impact of this liver subtypes regulation by the glucagon. Whenever you are fasting, in the fasting state, there will be a low glucose concentration, right, because you are not getting glucose from the diet. So, at that time glucagon hormone is synthesized. This glucagon hormone in the liver only, what it does, it inhibits this pyruvate kinase is inactivated. So, this step is inhibited and we can say glycolysis is inhibited. So, it inhibits glycolysis, right. So, what happens in the fasting, the liver cell stop utilizing glucose for the glycolysis. So it is sparing glucose. It is sparing glucose. Liver is sparing glucose. But for what purpose? This spared glucose can be transported to the other essential organ like brain and other tissue, so that this essential tissue does not feel that there is a low glucose concentration. So this completes our regulation of glycolysis. In the next video, I will talk about the applied aspect of the glycolysis. If you have any query or confusion, please write it down in the comment section below. Thank you.